Hello everybody. So today's topic which I am going to talk about is morphology of bacteria. Very important where your exam is considered, and yes, you should know the basic approach of morphology of bacteria. So let us have a look at it. What does it consist of? So in this topic, I will be telling you about the introduction part, size of the bacteria, shape of the bacteria, arrangements of the bacterial cell, structure of the bacterial cell. So let us go with the introduction part. Now bacteria is unicellular, consists of one single cell. So it is unicellular, free living, microscopic microorganisms capable of performing all the essential functions of life. They possess both deoxyribonucleic acid, which we call it as DNA, and ribonucleic acid, which we call it as RNA. Bacteria are prokaryotic microorganisms that do not contain chlorophyll, so they are not able to produce their own food. They are totally dependent on the host. They occur in water, soil, air, food, and all the natural environment. They can survive extremes of temperature, pH, oxygen requirement, the atmospheric pressure. Next is the size of bacteria. Now bacteria are very small. We require microscope to see them. They are visualized under the microscope. So they are that tiny which we can't see through our naked eye. Now they are having a size range in microns. Bacteria are stained by staining reagents and then visualized under a high power of magnification. We have to use 100x power to visualize it in a compound microscope. An electron microscope is used to clear the visualization of the internal structure of the bacteria. Shapes of bacteria, very important, you should know this. On the basis of shape, bacteria are classified under 7 categories. The first one, cocci, second one, bacilli, third one, vibrio, fourth one, spirilla, fifth one, spirochetes, sixth one, actinomycetes, and the seventh one is mycoplasma. Now let us see in each one in detail. The first one, cocci. Now cocci are small, spherical or oval cells. You can see the picture over here. They are small, spherical or oval cells. In Greek, coccus means berry, something similar to that of a berry, a fruit berry, which is round or oval in structure. Example you can remember here is Micrococcus. Second one, bacilli. Now they are rod shaped. You can see here they are rod shaped. Example is Bacillus anthracis. It is derived from Greek word bacillus, meaning stick. In some of the bacilli, the length of the cell may be equal to the width. Such bacillary form are known as cocobacilli. It gives appearance of cocci also, that's why it is called as cocobacilli. Example could be brucella. The third one is vibrio. They are comma shaped curved rods. Example is vibrio cholerae. Fourth one, spirilla. They are longer, rigid rods with several curves or coils. You can see exactly Maggi noodle appearance they have. So they have a helical shape and a rigid body. Example is Spirillum rubrum. Fifth one, spirochetes. They are slender and flexuous spiral forms, just like a spring appearance it is. Actinomycete. Now actinomycete, it is called as true filamentous bacteria. Why? The characteristic shapes is due to the presence of a rigid cell wall. Example is streptomycetes. Now they have branching filamentous bacteria, or they are called as branching filamentous bacteria. You can see they are just growing like a tree. Example, Streptomyces species. When you observe these actinomycetes under microscope, you will see them has a beautiful sun appearance or a beautiful flower appearance. Exactly similar look they will show. Mycoplasma. Now they are cell wall deficient bacteria. and has got no proper stability of morphology. Whichever form it is growing, it will take its shape. They occur as round or oval body with interlacing filaments. Yes, you can see they are with interlacing filaments. Arrangements of bacterial cell, yes. Now, cocci appear as a several characteristic arrangement or grouping, which we call it as diplococci, streptococci, tetracocci, staphylococci, and sarcinae. Now, coming to diplococci, yes, you can see their picture, a very beautiful way of understanding it. They split in one plane and remain in pair. They are attached together. See? They are present in pairs. Example is Diplococcus pneumoniae or Streptococcus pneumoniae. Second one is Streptococci. Now these cells divide in one plane, but they are remain attached to form a chain. Like this, they form in a chain. Example is Staphylococcus aureus is not the one. It is Streptococcus lactis. Now Streptococcus family will show you cocci arranged in chains. So do not get confused. Staphylococci will show you other formation. Streptococci will only show in chains. The third one is tetracocci. Now tetra means 4. Cocci means rounded structures. So they are divided in 2 planes and live in group of 4. Okay. Example could be Gaffica tetragena, Micrococcus. The fourth one is Staphylococci. Now cocci cells divide in 3 planes, X, Y and Z axis. Now in an irregular pattern. Now these cells will reproduce, or it will produce bunch of cocci as in grape-like clusters, which we call it as. Example is Staphylococcus aureus and Staphylococcus albus. Sarcinae. Now Sarcinea, instead of 4, they will produce 8 cells. Now see here, Sarcinae cells will divide in 3 planes in a regular pattern, and these cells will produce a cuboidal arrangement of group of 8 cells. Example is Micrococcus tetragena. Next one is arrangement of grouping formed by the bacilli species. It could be diplobacilli, where you could see 2 pairs of bacilli, streptobacilli, bacilli in chains. It could be trichomonas, yes, like this, in a filament form. It could be palisade, which is in parallel form. So let us have a look at the bacterial structure. Very important, it's a 5 marks question to be known. You should draw a beautiful diagram of a bacterial structure showing all the parts which is required. Yes, let us go with the parts. Flagella, you can see the tail-like structure here. Yes, that's called as bacterial flagellum or flagella. Pili, capsule, plasma membrane, cytoplasm, cell wall, ribosomes, mesosomes, inclusions and spores. So let us go with each one in detail. Yes, let us go with flagella. Now flagella itself is a 5 marks question, very important, where your exam has considered. So let us go with flagella. Now flagella are long, slender, thin, hair-like structure there, and cytoplasmic appendages. So remember, it is a locomotory organ of bacteria, now which are responsible for the motility of the bacteria, for the movement of the bacteria. Now these are the organs of locomotion. Now they are about 0.01 to 0.02 micrometer in diameter, 3 to 20 micrometer in length. Now flagella are made up of a protein called as flagellin. The flagellum has 3 basic parts, very important, you should remember. First one, filament. Second one, hook. Third one is the basal body. Yes, this diagram is important. It should be there in your answer sheet if you are writing an answer for flagella. So let us go with what is filament. Now filament is a thin, cylindrical, long, outermost region with a constant diameter. Now filament is attached to a slightly wider region called as hook, or you can tell a neck of the flagella. The basal body is composed of central rod inserted into series of rings. Yes, you can see here we have got L, P, S and M ring. Now, how does it vary with gram-positive and gram-negative? Gram-negative bacteria contains 4 rings. All these 4 are present. L ring, P ring, S ring and M ring. Whereas gram-positive bacteria, they have only S and the last one, M ring, in the basal body. They do not have the first 2 rings. But in gram-negative, you will see all the 4 rings are present. Next is the flagella may be seen on the bacterial body in a following manner. So let us go with the types of flagella which we see. The first type is called as monotrichous. Yes, you can see here. Now these bacteria have a single polar flagellum, one single flagellum attached. Example is Vibrio cholera. Second one, lopotrichous. Lopo, at one end it is attached. So you can see where 2 or more flagella is attached. Example is Pseudomonas fluorescens. Third one, Amphitrichous, that is this one. These bacteria have a single polar flagella on both the poles. Example is Aquaspirillum serpens. Peritrichous, several flagella present all over the surface of the bacteria. You can see everywhere they are present, all over the body of the bacteria. Example, it could be E. coli, which we call it as Escherichia coli, Salmonella typhi, all this will come. Next one is pili or Fimbriae. Now pili, it is a hair-like microfibril, much smaller than that of your flagella, and the number is more than that of flagella. So it is about 0.5 to 2 micrometer in length, 5 to 7 nanometer in diameter. They are thinner, shorter and more numerous than flagella. They are present only on gram-negative cells. They are composed of a protein known as pylene. In flagella, they are composed of protein called as flagellin, whereas in pili it is pylene. Now they are unrelated to motility and are found on motile organism as well as non-motile organism. Now pili and Fimbriae, these two terms are used interchangeably, but they can be separated or they can be distinguished. How? See, Fimbriae can be evenly distributed over the entire surface of the bacterial cell, or they can occur at poles of the bacterial cell. Each bacteria will possess about 100 to 200 Fimbriae. Pili are usually longer than that of Fimbriae, and the number is only 2 or 1 per cell. Now coming to function, pili play an important role in attachment to surface. Hence, it is also called as organ of adhesion, organ of attachment, organ of adhesion. The next important one, which we call it as cell membrane, cytoplasmic membrane or plasma membrane. So let us go with the plasma membrane. The cytoplasmic membrane is thin. It is about 5 to 10 nm. It is separated from the cell wall as well as cytoplasm. It is separated from the cell wall as well as cytoplasm. You can see here. Yes, this is a cytoplasm and this is a cell wall. So it is a separate unit which we call it as plasma membrane. Yes, the white structure. Yeah. Okay. Now it is composed of phospholipid 20 to 30%, protein 60 to 70%. Now prokaryotic plasma membranes are less rigid than the eukaryotic membrane due to lack of sterols. Now what is the function of this plasma membrane? It is semi-permeable, means it is allowing only thing which it is needed for the metabolic function of that bacterial cell. So it is a semi-permeable membrane controlling the inflow and the outflow of metabolites to and from the protoplasm or the cytoplasm. It provides the mechanical strength to the bacterial cell. It helps in DNA replication. It contains enzyme like permease, which plays an important role in passage of selective nutrients through the membrane. The next important one is cytoplasm. Now, bacterial cytoplasm is a suspension of organic, inorganic solute in a viscous water solution. Something like a jelly appearance it has got. A viscous water solution. Okay. Cytoplasm of bacteria differs from that of a higher eukaryotic microorganism. Now, if you say, take example, an animal cell. Now, animal cell or any eukaryotic microorganism, you can see it will have its own cell organelles like endoplasmic reticulum, Golgi apparatus, mitochondria or lysosomes. But in bacterial cells, we do not have any of these except ribosomes. Okay, so remember, now cytoplasm of bacteria differs from that of higher eukaryotic microorganism in not containing endoplasmic reticulum, Golgi apparatus, mitochondria and lysosome. They are absent here. It contains only ribosome, proteins, other water soluble components and the reserve material. In most bacteria, extra chromosomal DNA is present, which we call it as plasmid, is present. The next one is mesosome. Now, mesosomes are the respiratory sites in bacteria. Now, mesosomes are attached. If you see any invagination, if you see the textbook reference of the bacterial cell structure, it will show you invagination inside. Something which has come inside the cytoplasm. Yes, that is what we call it as mesosome. Now, mesosomes are attached to the bacterial chromosome and is involved in DNA segregation. Helps the DNA to cell divide or cell division will take place. Now, they are predominant in gram-positive bacteria. Nucleoid. The bacterial chromosome is not surrounded by a nuclear membrane, so it is called as nucleoid. The bacterial chromosomes are made up of double-stranded circular DNA. Intracytoplasmic inclusion. Now, many species of bacteria will produce some particular inclusion body, which we call it as cytoplasmic inclusion bodies, which will appear round granules. They are round in structure and they are made up of purely glycogen or starch. Now, they will appear as reddish when stained with polychrome methylene blue or toluidine blue. Next one is capsule. Yes, capsule is very important. It could be a 3 marks question to remember. Yes, let us go with it. Capsule. A bacteria synthesizes a loose amorphous organic exopolymer which is deposited outside and it is tightly attached to the cell wall, which we call it as capsule. Now, you can take it as a rubbery structure just covering outside the bacteria, which helps bacteria from protecting with so many things. Now, capsule may be composed of a complex polypeptide or a polysaccharide, but the main component of this bacterial capsule is water. 98% is purely water. Sometimes the capsular material is so loosely present, okay, it could be easily washed away with the water. This loose layer, we call it as slime layer. I am sure as a child you must be playing with that slimy gel, no? Yes, exactly, same appearance you will have. Now, this capsulated bacteria produces a smooth colonies and a non-capsulated bacteria will produce rough colonies on the agar surface. Yes, on agar media. Now, coming to the function of it, yes, you can see that it protects the cell from drying because it is rich in water, so it gives all the nourishment and the well-grown bacterial cell. Now, they protect the bacterial cell against antibacterial agents and helps it from phagocytosis. Cell wall is very important. Cell wall is a rigid structure which gives a definitive shape to the cell, situated between the capsule and the cytoplasmic membrane, which we call it as plasma membrane. It is about 10 to 20 nanometer in thickness and constitute about 20 to 30% of the dry weight of the bacterial cell. Now, the cell wall cannot be seen by direct light microscopy and does not stain easily by different staining reagents. The cell wall of bacteria contains diaminopleamic acid, which we call it as DAP, and muramic acid and teichoic acid. Now, if these substances are joined together, and it gives rise to a complex polymeric structure which we call it as peptidoglycan or murin or mucopeptide. So they are consisting, the group of substances DAP, muramic acid and teichoic acid forms a peptidoglycan. Now peptidoglycan is a major constituent of the cell wall of gram positive bacteria, which is 50 to 90%, whereas in gram negative they are very little. Okay, so bacterial cell wall, its presence is only 5 to 10%. Yes, you can see this is how the chemical structure of the bacterial cell wall consists of N-acetyl muramic acid and N-acetyl glucamine attached together by a peptide chain, and you can see, yes, it could be by pentapeptide chain. So let us go with a comparison of the cell wall of a gram positive and gram negative bacteria. It is important. It's a 5 marks question. You should know how you differentiate between a gram positive bacteria and a gram negative bacteria. So let us go with the first one. The first side, you can see on the left side it is the component, and then it is gram positive, and then it is gram negative organisms. So let us go with the first point, thickness. Gram positive, it is thicker, 20 to 25 nanometer in size. Gram negative, it is thinner, 10 to 15. Peptidoglycan, yes, as I told you before also, it is more in gram positive, which is about 50 to 90%, whereas it is less, it is 5 to 10% in gram negative. Teichoic acid, yes, it is present in gram positive, absent in gram negative. Polysaccharide, again present in gram positive, absent in gram negative. Lipids, lipid is absent or less in gram positive, but it is more in case of gram negative. Cell wall, simple in gram positive, but it is complex in gram negative. Outer membrane and the periplasmic space is absent in gram positive but present in gram negative. Effects of lysozyme, easily destroyed in case of gram positive, resistant in case of gram negative. Types of amino acid, very few, but here you can see it is several. Susceptibility to streptomycin and tetracycline is slightly positive, but you can see a very well marked susceptibility in gram negative. Susceptibility to antibiotics like penicillin and sulfonamides, yes, it is effective in gram positive but very less in case of gram negative. Examples, yes, you can remember gram positive organisms like Bacillus anthracis, Clostridium tetani, Staphylococcus aureus, Corynebacterium diphtheriae, whereas gram negative, Escherichia coli, which we call it as E. coli, Salmonella typhi, Vibrio cholerae, Haemophilus influenza. Let us go with the function of cell wall. Cell wall is involved in growth and cell division of bacteria. It gives shape to the cell. It gives protection to the internal structure and acts as a supporting layer. To prevent rupture of bacteria caused by osmotic pressure, differences between intracellular and extracellular environment. To provide support for flagella, because flagella is coming out of the cell wall itself. To regulate the central certain degree of passage of molecules into and out of the cell. It serves as a site of attachment for most bacterial viruses. The next one is ribosome. Now ribosomes are the center of protein synthesis. Yes, you must have studied in your +2. Now they are slightly smaller than that of eukaryotic ribosomes. The sedimentation constant is 70S. Now S stands for the sedimentation constant. You can see these 70S ribosomes are made up of 2 subunits, namely the largest subunit which we call it as 50S and the smallest subunit which we call it as 30S. Now during active protein synthesis, the ribosomes are associated with mRNA, and such associations are called as polysomes. Now these mRNA and ribosome attaching together will form individual amino acids, and they form a chain to form a protein. The next important one, yes, it is a 5 marks question. You should know what is a spore or endospore. Now the process of endospore formation is known as sporulation. It may take about 4 to 8 hours in a vegetative cell. Endospores are thick-walled. They are very thick, highly refractile bodies that are produced one per cell. Now, what is the characteristic in this endospore? See, there are certain bacteria during an unfavorable environment go to a sleeping stage for about thousands of years time. Now that sleeping stage is called as endospore. Now each bacterial spore on germination will form single vegetative cell. Therefore, sporulation in bacteria is the method of preservation and not reproduction. Now spores are extremely resistant to desiccation, staining, disinfecting chemicals, radiation and heat. They can all resist this. They remain viable for centuries and help bacteria to survive for a longer period of time under an unfavorable environment. Now, endospore can remain dormant for thousands of years. Now, spores of all the medically important bacteria are destroyed by moist heat sterilization, that is autoclaving, at about 121 degrees Celsius for 20 minutes. Let us go with the bacterial sporulation steps. You can see once they have come activated, a vegetative bacterium will undergo replication process. So DNA will replicate. Septum is formed. Now the DNA will translocate into a spore. You can see here. Now spore is engulfed. The spore forms a thick coat outside. Then finally the mother cell will break or lyse to form, you can see, it will form an individual endospore. It is released out. Yes. So that's it for today, and I hope it is clear and it is understood for all of you, the morphology of bacteria. And yes, if you are a new joinee, please do subscribe my channel and make it a hit. Thank you so much, and see you in my next topic, which could be about physiology of bacteria. Thank you.